Afternoon. My name is Cleopas Cleopa. Thank you for joining this webinar on gene therapy developments. This is actually the first webinar in a series that we have planned that will take place from today until July. We have 12 webinars planned. This is an initiative of the Inter-ERN Working Group on Gene Therapy. This is a joint working group between the European Reference Networks on Neuromuscular Diseases, Rare Neurological, and Epilepsy, EpiCare. And we would like to cover basic aspects of gene therapy, challenges in the development of new gene therapies, update clinicians and scientists on the recent developments, both in terms of the preclinical, but also in the approved therapy. So we have some excellent speakers that will present the webinars, and would like to welcome you. Again, this is also in partnership with the European Academy of Neurology. And we will start today with two excellent speakers. And I will go ahead and introduce our first speaker for today. Is Dr. Munell is Dr. Francina Munell Casadesus from the Hospital Vall in Barcelona. Dr. Munell is a pediatric neurologist working in Barcelona and coordinator of the Pediatric Neuromuscular Unit. Her main interest is the study of early onset neuromuscular disorders. Professor Munell participates in clinical trials using gene therapy for spinal muscular atrophy and Duchenne muscular dystrophy, and also in research projects aiming to develop a therapeutic strategy for laminin muscular dystrophy, and in the elaboration of a guideline for gene therapy in neuromuscular disorders in Spain. Dr. Munell will talk about the general overview of gene therapy developments for clinicians. And I would just like to note that you can enter your questions during the talks and in the question and answer session, and we will have the question and answer session at the end of both presentations. So with this, thank you again for joining, and Dr. Munell, please go ahead with your presentation. Thank you. If you please unmute.
Okay. Do you see the presentation?
Yes, looks fine.
Okay, thank you. Well, hi everyone, and thank you for inviting me to deliver this presentation on gene therapy. And this is our team in pediatric neuromuscular disorders, 4 pediatric neurologists and 1 nurse, and they have been involved in the clinical trials and also treating patients in real life. Well, as you all know, gene therapy is now a reality. Using the words of Jerry Munell, gene therapy greatly improves the outlook for currently incurable inherited diseases. And the clinical trials that have been developed in the last years demonstrated the efficacy and the safety for some of these therapies. And now, we have 2 therapies in real life in neuromuscular disorders. This is the SMA in Europe and United States and other countries, and the DMD by now only in the United States. But we have a lot of studies involving gene therapy, an increasing number of studies every year, and in neuromuscular disorders we have also clinical trials for some limb-girdle muscle dystrophies, or the sarcoglycanopathies, for Charcot-Marie-Tooth disease, for giant axonal neuropathy, and others. What is gene therapy? Gene therapy is a therapeutic approach that uses genetic material to prevent, treat, and potentially cure a disease. We have different key approaches. We have the gene replacement therapy that is the most used now, that consists of the replacement of a mutated gene by a healthy functional gene. But we have also the gene addition approach that here we use the therapeutic gene only as a supplement to a specific therapeutic agent. We have also the gene inhibition. When we inactivate a mutated gene, usually a mutated allele that is overproducing a product, a mutated product, and we do this inactivation by working on the RNA produced by this mutated allele. And finally, we have also the gene editing, that here we will not replace the mutant gene, but we will try to correct specifically the mutation. We have also to know that there are two types of gene therapy, the in vivo and the ex vivo gene replacement therapy. In the in vivo therapy, a single dose administration of the viral vector, this viral vector that will contain a nucleic acid sequence composed by the cDNA of the gene of interest, that we will call it transgene, and the promoter. And we will administer this viral vector once, usually by intravenous administration. In the ex vivo gene therapy, what we do is to isolate the cells, in this case will be hematopoietic cells, and we'll do the transfer of the gene and transduction of the cells outside of the body of the patient, and then we'll reinfuse the gene modified cells, in this case without the virus, the vector virus. In the following slides, I will explain all of the actors involved in gene therapy. The viral vectors are naturally occurring viruses that have been modified. Most of the original viral genes of the virus have been modified, and here we have in the vector for gene therapy, the promoter and the transgene. These ITR sequences, the inverted terminal repeated sequences, will be maintained on this DNA because they are the origin of replication, are important sequences for the synthesis of cDNA and expression. And removing these rep and cap genes of the original virus, the virus will be unable to replicate and trigger the same immune response of the wild type virus, and we will increase the efficacy and the safety. The ideal virus will have enough sites to accommodate the gene of interest, will have an efficient distribution and delivery. We will try to accumulate the virus or the viral vector in the tissue and cells of interest and try to avoid the other tissues to increase safety. It will be non-immunogenic and non-inflammatory. That is not possible, but we are talking about ideal vectors. We will need to have high titers of the vectors to have enough expression and a maintained expression over time, and it will be safe for patients and environments. Some viral vectors have some of these characteristics, but they are not ideal vectors. And this is why they are under investigation. Non-viral delivery systems, but the ones that we have now, they have poor efficiency and low and transient expression of the transgene. The viral vectors used in gene therapy can be integrative and non-integrative. The integrative vectors integrate into the DNA of the host, of the patients, and are, for example, the retrovirus and the lentivirus, and the non-integrative persist in the cell as episomes outside the DNA of the cell. And these are the three main non-integrative viruses. Each of these type of virus will be used depending of the type of tissue. For proliferative tissues at the blood cells, we will need to use in integrative virus because the episomes, the non-integrative sequences, are progressively diluted through cell division. And if the rate of proliferation is high, we will lose this virus and the cell. But in non-integrative cells, as neurons, we can use the non-integrative virus and the AAV virus and any post-mitotic tissue. These are 4 types of episomal viruses. The lentivirus has some percentage of integration. As you can see here, the size of the virus is very different. We have these large big viruses, the adenovirus and the herpes simplex virus, that have the advantage of a high clonic capacity because of their size, but they are highly immunogenic, and the production is highly complex. We have this lentivirus that is not the optimal virus for gene therapy because in vivo gene therapy, because as I said, have some percentage of integration, but it has moderate in vivo efficiency. And the preferred one is the adeno-associated virus because it's highly safe for patients, but have this problem of the limited clonic capacity. Then the AAV virus are optimal for in vivo gene therapy. These are DNA virus, non-pathogenic with limited immunogenicity, can be produced at high titers, and can cross endothelial barriers at the blood as the blood-brain barrier. To face the cloning capacity of this virus, we can reduce the size of the therapeutic gene, and this has been done, in hemophilia A by deletion of the F8 domain, this B domain, and also in Duchenne muscular dystrophy, and as you all know, creating these mini dystrophins and microdystrophins that, although they are not, they do not produce the full protein, the protein is still functional. This is the whole procedure of the cell transduction by these AAV vectors. The vector with the transgene bounds to the cell membrane and is taken up into the cell via endocytosis, a clathrin-mediated endocytosis, and the endosome is formed, the endosome matures, and finally breaks down, and the complete viral particle, the virion, is released into the cytoplasm that will be ubiquitinated and degraded through the proteasome. But part of the viral particles will enter the cell nucleus, and the DNA is where the DNA is released, the uncoated DNA, and then this DNA will fold upon itself to form a cell complementary double-stranded DNA molecule that is the episome. The episome is ready for transcription, and the transcript will leave the nucleus and travels to the ribosome for translation into the therapeutic protein. Now we can say that the cell is functionally transduced. To increase the efficiency of the distribution, we can play with the promoter and also with the serotypes or pseudotyping of the viral vectors. We have some natural pseudotypes of vectors with antigenic differences in the viral capsids that recognize specific receptors, sometimes they use also co-receptors of the cell membrane. And for example, as you can see here, we have the AAV8 and AAV9 that have a trophism for the skeletal muscle and also for brain, but also for liver. And this will be a problem because we will have hepatotoxicity when we will use these serotypes of AAV. Then novel capsids and tissue-specific promoters will be needed and are under investigation to increase this tropism and also the safety and efficacy. We also can choose different routes of administration. The most used is the intravascular, can be intravenous, intracarotid, or intrafemoral, but also we can use the delivery directly to the central nervous system, different parts of the central nervous system, and also intramuscular. And now we will focus on the host. There are natural immune barriers to these AAVs because a high percentage of the general population, not treated persons, have high titers of neutralizing antibodies against AAVs. This seroprevalence is around 30 to 50% of the general population, and this is age and population dependent. We saw this when we started the trials, the clinical trials using AAVs, and we saw that it was not a problem for babies with SMA, but for boys aged 4 to 8 with Duchenne muscular dystrophy. At least in Spain, 50 of them were excluded from the clinical trial because the presence of these neutralizing high titers of these neutralizing antibodies. After the treatment, after the systemic administration, all of them will develop neutralizing antibodies. And for this, we need some treatments to decrease, at least transiently, this level to be able to treat the patients that were excluded from the trials, but also for patients that have been treated, because we don't know in some years they will need a second dose of treatment. The host also will have some reactions, immunoreactions against the AAVs, the ones treated. No, these are frequent reactions that usually we can treat without problems with the dose of corticosteroids that we give them preventively. That is the hyperthermia, the vomiting, the loss of appetite, or the dehydration. We will also see an increase in acute phase reaction, that is, for example, an increase in ferritin and an increase in albumin, and also cytopenia, usually a decrease in platelets, but usually with corticosteroids it is enough. This should usually is transient, and other treatments are not needed in the majority of patients. We can have also reactions in specific tissues. In the liver with hepatitis, we will see in most patients increase in transaminases. Usually we have two peaks at different moments of increase in transaminases, And with corticosteroids, sometimes increasing the dose of corticosteroids in most patients, we can treat this problem. And also myositis and myocarditis with the increase of CKs and troponin I. And in some patients, we will have to perform electrocardiograms or MRIs to know how big is the problem. Another reaction of the host is the complement activation, with the appearance of a microangiopathy and an atypical hemolytic uremic syndrome. Again, this has an age-related frequency. It has been more frequently seen in boys with Duchenne, and because of the age. And the presentation is usually progressive in a few days, but we need an early detection and treatment. Then we will have to check the schistocytes, the blood pressure, the presence of hematuria, to be able to detect this problem as soon as possible. And in some cases, we will need to treat the patients with eculizumab. We will have to have these medications always in our hospital to treat this possibility. Some adverse events will depend of the condition. For example, in the myotubular some patients have liver problems, and after the treatment, this liver inflammation led to 2 deaths in these Aspiro clinical trials. This is why it's so important to know how is the patient before the clinical trial. Also, we can have long-term problems. We know that human gene therapy products may be associated with delayed adverse events because the gene therapy product could be integrated into the host genome. We said that these AAVs are non-integrative virus, but can be integrated in a very low percentage, or because of the prolonged or excessive expression, because this is a non-regulated expression, it's a high by non-regulated expression. The experiments done in some big animals, for example, in the model of in dogs of in hemophilia A, it has been seen at 10 years, this is a publication of the last month, that the vectors persist in the liver of these animals as an episomal forms after these 10 years, and there is vector integration at very low frequencies, but occurred in areas of open chromatin with no effect on gene expression by now. But we need long-term clinical studies for monitoring the effectiveness and safety of these treatments. Then the conclusions of this first part of the talk are that gene therapy is a clinical reality. The indication to be treated, the route of administration, and the vector design are the critical factors related to the clinical safety and efficacy. The adeno-associated virus are the most used vectors, but novel capsids and tissue-specific promoters should be developed to increase safety and efficacy. That there are immune barriers to AAVs with a high seroprevalence of neutralizing antibodies that are age and population dependent, and that are short-term concerns of safety, and it will be important to do pharmacovigilance in all these treatments in real life, because we have immunoreactions against the AAV and also the complement activation. And we could have also long-term efficacy and safety problems that by now remain unknown, but we need long-term studies to know. And thank you.
Great. Thank you very much, Francina, for this excellent presentation. That was a really nice coverage of the basics of gene therapy and also some clinically very relevant issues and questions that we face. For the colleagues who joined later, just a reminder, you can enter your questions and we will have the question and answer session at the end after the second talk. So we will now go to the second part and to the second presentation by Professor Laurent Servais at the University of Oxford. He will talk about gene therapy in clinical practice, when and how. Professor Servais is a pediatric neurologist and works at the Neuromuscular Diseases unit in Oxford. He's also an invited professor at the University of Liège in Belgium. He graduated in medicine and pediatrics from the University of Louvain in Belgium and trained as a child neurologist in Paris and as a myologist at the Institute of Myology at Salpêtrière in Paris. His main research interests cover innovative outcome measures and clinical trials design, as well as newborn screening. He has been involved as principal investigator in several clinical trials in spinal muscular atrophy, Angelman syndrome, X-linked myotubular myopathy, and Duchenne muscular dystrophy, and in leading the NBS program for SMA in Belgium and the UK. He is the coordinating investigator of two large natural history studies in Angelman in the UK and Belgium that aims to identify and validate innovative outcome measures and biomarkers. So he has a lot of experience that he will share with us today. And thank you again for accepting. And we look forward to your talk. Thank you.
Thank you so much, Cleopas. I was quite in trouble because Francina sent me a slide some days ago, and then I said, what I'm going to tell them after that. I mean, it was quite complete, so I felt that maybe what I could add on top of when Francina presented was to take one or two steps back and say, well, you know, what are the other issues that we could be facing, and just believe me, I don't want to be negative. I'm just going to try to say, well, okay, what could also be the problems? So the first thing is that I have a lot of conflict of interest. It's my disclosure. And I'm going to specifically consider the case of two diseases because I'm quite a narrow-minded person. So I would discuss the disease I know quite well, which are SMA and Duchenne. And I think there are two very good examples to bring, you know, some thinking about the limitation and the challenges that we are facing. So spinal muscular atrophy is a disease of the motor neurons. Duchenne is a disease of the muscle cells. The phenotypic spectrum in SMA is very broad. It starts with baby and it can also affect adults. Duchenne is more homogeneous on boys, and they, you know, of course there are several phenotypes, but yeah, it's quite a little bit more homogeneous. And then the size of the transcript is very different. It's a very small transcript in SMA and much longer in Duchenne, so no way to put this giant, which is dystrophin, into a teeny AAV. The type of mutations is very different. It's always the same for SMA. It can be any type of mutations in Duchenne. And it's a multi-tissue disease, the Duchenne, not so much for SMA. It's mostly motor neurons, right? And the incidence is pretty the same. So Duchenne muscular dystrophy. Let me show you a video of a patient who has been treated. You can see on the right side the treated kids and on the left side before the treatment.
Vas-y Wilson, sur la rampe, hein. Oui, ouais, super, continue, c'est bien, continue, c'est bien. Encore, ok, sur la rampe. Même chose que oui. Eh bien, okay.
Bravo. So you can see it works beautifully, right? But actually it's not gene therapy, it's steroids. So we gave steroids to the patient. And the reason why I show it very provocatively is that, one, we have standard of care for Duchenne, and we should really think about first applying standard of care before giving innovative therapy, but B, do not forget in the clinical trials that we give steroids on top of gene therapy, and that steroids works very well. So when we have to interpret the data of open label study in Duchenne, we need to keep in mind that we give also steroids and high dose of steroids to these patients, and that the steroids, they may work pretty well. So just to say that we need to apply good standard of care, and I'm not going to give a lecture on standard of care in Duchenne, but it doesn't make sense. It doesn't make sense to think about gene therapy and whatever it is before applying good standard of care. And again, let's be careful to steroids effect. This is an excellent paper that demonstrated in 4 kids the proof of mechanism and the target engagement of AAV construct microdystrophin. But when it comes to the clinic, and we see that these patients improve beautifully, like 9 points on the North Star. 9 points is big, it's massive. We need to keep in mind that these patients are quite young. There is a maturational effect, and also we give a high dose of steroids to these patients. It means that it's difficult to compare them with natural history study, because in the natural history, we don't give all of a sudden massive amount of steroids. And this might explain why when this phase 1 study moved to a phase 2 studies that was blinded, we could not observe this magnitude of effect, right? It was not a question of 9 points on the North Star anymore. It's a question of maybe 1 point, 1.5 points. And this was the phase 2. It was not statistically significant, but it was a small study. And actually, it seems that the effect is probably more obvious in the younger patients. But again, we're not talking about 9 points anymore. We're talking about 2 points, right? So again, let's keep in mind that when we go to a double-blind randomized placebo-controlled study, things may be a little bit more complicated. And then we had a phase 3 trial that was actually on the primary endpoint, the North Star, unfortunately not a success. So we were very far away of the 9 points on the North Star, but on several outcome, the secondary outcome, there was a difference. But again, let's keep in mind that patients who received actually gene therapy in comparison with patients who received placebo could have received more steroids because of the side effects. Because the first thing that Francina and myself, we do, if we see a patient after gene therapy with vomiting, with doing unwell, we keep this patient in the hospital, we place an IV line, and we just increase the dose of steroids for quite a while. So we need to be careful of this steroids effect. We need to be careful also that the gene therapy is a very exciting approach, but it comes with a risk, right? It comes with a risk on the heart, and you've probably heard about what could happen in one of the gene therapy trial. And recently, this paper in the New England Journal of Medicine reported on a fatal case. To be honest, the patient was 27 years old, which is quite old to go for a gene therapy. But nevertheless, the patients died after obviously a multisystemic inflammation that was probably related to an immune reaction. The problem we will face in the Duchenne is the timing, and this is an interesting feature in comparison with SMA. In SMA, we will want to treat as soon as possible. In Duchenne, it's not that clear, because if you treat very, very early on, you will have a dilution. The dose that you give to a patient with 3 kilo will be obviously lower than a patient with 60 kilo. But if you weigh 60 kilo, then the patient has no muscle anymore. So there is a sweet spot somewhere, and the problem is that nobody knows exactly where is the sweet spot. And the second point is that probably the sweet spot is not at the same moment for the different patients. If you have a very aggressive disease, maybe the sweet spot is much earlier on. It brings me to spinal muscular atrophy. And the first thing to know about spinal muscular atrophy is that, yes, we have gene therapy, but we have other medications that are approved. And that's going to bring additional challenges. So we have also oligoantisense nucleotide, and we have also a small molecule that interacts with gene therapy, with SMN2 splicing. This is the vector, and this is the construct. And look, this was the first paper published in 2017. Wow. It's wow. I mean, we just see patients with the high dose who are doing so much better. Well, actually, is it the high dose or the fact that they were treated early? It's not entirely clear, and we will never know, because the patients with the low dose on the left, they're not doing so well. They're surviving. They're not doing so well. But this being said, if you look, they have been injected at between the age of 6 and 8 months, just like the patients who was injected with the high dose and was not doing well. So was it an effect of the dose or the age of injection? We will probably never know. Nevertheless, just to show you two examples of kids who have been treated with spinal muscular atrophy, I used to present two kids, one from Belgium, which is a country where we have good beers and good chocolates and good fries and good waffles, and another one from another country. And these two kids have the same disease, right? And they've been treated with the same drug in the same hospital. But you will see that there is a big difference between them. Here is the little French guy. The little French guy is, after 1 year, he could survive. He kept a very, he doesn't need a non-invasive ventilation. He is a little bit limited with the upper limb, but he's doing very well, of course. He became a painter. He became a painter, but a painter with some limitation of the upper limb. And the good news is that now he goes to school and he's an happy kid, but he will never walk, right? He will never walk because very, very few patients who are treated after the symptoms actually walks. But this is the Belgian kid. He became a neurologist. He did not become a painter. He became a neurologist, and a very good neurologist, as you can see, because he takes very well the reflexes of the patients. But the difference between these two kids is not the Belgian fries, it's not the chocolate, it's not the waffles, it's even not the beers, it's the fact that this kid was identified at birth by newborn screening in southern Belgium. So for SMA, gene therapy is a fantastic game-changing drug, but too late, then the effect exists, but it's not. That's true for Duchenne and SMA. We see patients coming from all over the world now, and we have to explain sometimes that, yeah, these drugs works well in patients who are properly followed up in good standard of care. Early treatment for SMA is absolutely key. For Duchenne, it's not that clear. We don't know exactly when we have to infuse, and the problem of the timing of infusion is a big problem in Duchenne. In SMA, it's crystal clear. You have to give it as early as possible. And then one-shot injection can be extremely attractive, but it comes also with some ethical and issues. And one of the issues related to one-shot injection is that some parents may believe that one shot and that's it. It means that you just have to make a single effort to get the magic fix and that's it. And if, of course, if it's not paid by the Social Security, then comes the problem of crowdfunding. In crowdfunding, I mean, we are facing many patients coming from different countries and willing to go for gene therapy after having been successful in crowdfunding. It brings very significant ethical issues, I can tell you. And for me, the main ethical issue is that we just put our kits on the market and people have to give money in general. And that has been clearly demonstrated that, for instance, the most successful are, for instance, in the United States, people from a white background, people from an educated background, male. So there is a huge, inequity in the success rate of crowdfunding. And for instance, in Belgium, we had a very famous case and within 3 days, the family raised 2 million. And it went on the news. Wow, the Belgians are so generous. They give 2 million in 3 days. Yes, but then my patients and they were told, but because there is a drug that doesn't work, which was a drug that cost a lot of money, but that works. But we want to give the drug that cures, which is the gene therapy. And then I can tell you, my patients in Liège, from a poor city, they decided also to try to raise the 2 million, and they organized a party and they sold, perfume and waffles and chocolates, and they could raise 5,000 euros. well done, but you need 2 million. It means that for the parents, it was the triple jeopardy. They have a they have a baby who is sick. They are told that there is a drug that is better than the one that they receive. And if they don't receive this drug, is because they are not strong and good and smart enough to raise the money. So it's the triple jeopardy, and 2 of the jeopardy are because of us, and because we let them believe that it's their role to find the money, and that if they had the money, they could be cured, which is not true. So we need to protect our patients and our patients' community against that. If you don't like crowdfunding, the bad news is that there is also the lottery. And there was a kind of world lottery to offer 100 doses of gene therapy worldwide, which is a very generous idea of offering gene therapy worldwide. But actually, it comes with also massive ethical challenges. And unfortunately, in some countries, the gene therapy was not properly delivered and follow up, and it ended up in patients who died. So I'm not saying that it's not a good idea. I'm just saying that we should carefully think about all these ethical questions that come, especially when we raise in the community the idea that there is a kind of magic drug. And then comes also some medical and economical issues. How do we actually reimburse a one-shot therapy? It's not that easy from a modelized perspective, especially the fact that a patient receives a one-shot therapy after symptoms, if it's SMA, but also I'm pretty sure Duchenne, will never be cured, will never be entirely symptom-free, which means that these patients will come back and ask for another therapy, which will also increase the cost. How are we ready as a society to organize that and to deliver not only these drugs, but also to consider the proper funding process? It's not entirely clear. And look, this is from the phase 3 of gene therapy for SMA. These patients do better. They have received a one-shot injection, but like this guy, they will never walk. So they will come back and ask for another medication, like for instance, nusinersen or risdiplam. And we know from registry that many patients who have received gene therapy are also on another drug. And it comes with clear economical questions. So we really need to manage the expectations at the community level. If we make the community believe that it's better than another care, especially if it's not true, then we need to be ready to see the patients coming and knocking the door to get the treatment, which is entirely normal. A very good example, for instance, are these 2 patients. They were told to be symptomatic. We know it's impossible because when you are 50, when you score 50 on the shop intent and you are 2 weeks old, it means that you have not been identified per symptoms. And we know that they have been identified by familial screening. The problem is that if you show the video of these kids, and this is an example, this kid is doing perfectly well. And it's fantastic. And it means that it works, because this kid should be dead in absence of gene therapy. The point is that it's not a real SMA type 1 patients who have been identified per symptoms. It's a patients who have been treated very early on because he was identified by family screening. But if we let the community believe that this is a reasonable expectation for patients with type 1, then of course the parents will sell their house, they sell their car, they will relocate, they will kill you to get access to gene therapy. Unfortunately, you don't get a similar results with the patients who have been treated after the symptoms. And this is also true for Duchenne. Now that the worms are out of the can box, when the family have seen these results with potential 11 points on the North Star, which is massive, now they are truly believing that it's the best possible cure under the sky, that the child could be cured, and we have many patients from Europe who are fundraising to get the drug infused in the US, because today gene therapy for Duchenne is not approved in the US. So again, I'm not saying it doesn't work. I'm just saying, well, 11 points is not the reality. So we need really to manage the expectations at the community level. And we need also to manage the expectations at the patient's level. When we start treating a patient, if we tell them, well, we have a very good drug, you have a severe condition, but you have a very good drug, it's going to work. of course, is going better, but if you don't give a reasonable expectations, at one point family will come back and say, well, it's not perfect. Now let's go for the extra mile. Let's push the envelope, right? When I have a patient with SMA type 1 nowadays, I say, well, this is a terrible condition. Your child has the potential to live. He has the potential to be happy. He has probably not the potential anymore to be a walker. So we need to be very careful and set realistic expectation at the community level, but at the patient's level. And then we need to know that sudden unexpected serious adverse reaction happens in the real world and are much broader than in the clinical trials. The best example is the TMA or the hemophagocytic the hemophagocytic lymphocytosis, which has been observed in the real world, but was never observed in trial for spinal muscular atrophy. And this comes with the perception of the risk. The patients and the doctors have a very different perception of the risk. If this is a patient, if I have a drug, and a drug that has a decent chance to improve my patients, but comes with a 10% chance of casualty, the patients could think, well, with this drug, I have 90% of chance to go better. And the doctor could say, well, if I treat 100 patients with this drug, I will kill 10 of them, right? So we think in terms of population when we think in terms of risk. Our patients, they think in terms of benefit. And sometimes it brings two different understanding of the ratio between the benefit and the risk of a drug. And finally, I would say, let's consider global inequity in the treatment access, and let's be ready to manage, especially if now everything is global. If I'm diagnosed with spinal muscular atrophy in Burkina Faso or in Sudan, I will know that drugs exist. And I will, if I've got a fighting spirit, I will wish to relocate and to go somewhere where I can go this drug. So we need to be ready for that. We face this issue on a daily basis with spinal muscular atrophy. I can tell you, we will face this issue with many other conditions. Of course, I don't think that crowdfunding, and I don't think that lottery is the way. I think that the way is to work right now with low and medium income countries and to consider that disease modifying therapy is just one part of the tripod. There are two other very important parts. The first one is standard of care, because without standard of care, again, it doesn't make sense to apply gene therapy, and also newborn screening when it comes to SMA, to be sure that we treat the proper patients and that we maximize the efficacy. So just to finish, I would like to thank my team in Oxford and in Liège, and I hope that this lecture will help to open the discussion. Thank you so much for your attention.
Thank you very much, Dr. Servais. That was an amazing presentation, and all the very important points about safety, about interpretation of efficacy, timing, and the ethical issues, of course. So I would like to thank again both speakers, Dr. Munell, Dr. Servais, and now we have some time for questions and answers. So I will go briefly over some of the questions we have on the Q&A box. So the first one was by James Sando, and the question is, for therapeutic genes, transgene within the AAV vector, I'm interested in knowing, where does the transgene come from? Does it come from cDNA obtained from some pre-existing genome? Is the gene built nucleotide by nucleotide in a lab using a known sequence, or from some other source? I think that's both of you can answer that.
Francina, you can take it if you wish. I think that it's actually different according to the gene and to the gene process, because as you might imagine, for the for instance, for the microdystrophin, it is a pure entirely reconstruct genes, right? So for SMA is a much smaller gene that you can directly sequence. So Francina, anything to add on this?
Francina, you're muted.
Okay. Now that we can build in a lab an oligonucleotide, but the whole gene is difficult. And then what we do is to clone the gene from a source of a human cell that we know that has a normal gene, and then we can cut the introns or the pieces that we need or we do not need and construct the transgene.
Yeah, so it's always a cDNA, essentially. It's not the actual gene because it has many non-coding areas. Thank you very much. I think that was clear. And the other question is, can AAV be related to Guillain-Barré syndrome? So it's an immune reaction to the gene
So
therapy.
I'm not aware of Guillain-Barré syndrome. What we have observed in intrathecal injection, but much more with antisense, is a polyradicular neuropathy. I'm not aware of Guillain-Barré syndrome as a potential direct complication of AAV gene therapy.
I'm not aware of that either. I
Yes.
haven't heard of this. Yes.
No, I'm not aware either. It was related with the adenovirus used for vaccines for the COVID, but I think that for adeno-associated virus in gene therapy, it has not been reported.
Great. Okay. So the other question is from Eugenio Zapata. What do you believe will be the future of NMD treatment disorders like SMA? Do you think the multiple drugs will be useful, gene therapy plus ASO, plus myostatin, MAGIC, or the gene therapy will be the only treatment available? I think Laurent may want to answer this.
Well, I think that the future is first a newborn screening. And when I say newborn screening, it's not only to implement newborn screening, but to do it as fast as possible, and to accelerate our process to treat as fast as possible these patients.
Yeah, I think
Big fan of combining all these drugs together on the same target.
we have some interruptions. Yeah.
So I was saying that can you hear me?
Yes, now yes.
Yeah, sorry for that. So I was saying that I'm not a big fan of using all drugs together that works on SMN because I'm not sure it works, but I'm sure it costs a lot of money. So I would say that for me the next step, if we want to push the envelope for patients who are already symptomatic, is one to change the target, and one of the target could be the muscle. In for instance with anti-myostatin, and also all the non-pharmacological therapies, including exercise, spine stimulation, also maybe to work on the neuromuscular junctions. I think we need to be honest with the community and the patients. I don't think that today we have the potential to cure a patient with SMA who is already symptomatic and treat it afterwards. We can potentially improve these patients a little bit more, improve the quality of life of these patients and the family, but we need to be realistic. And again, the future is probably to try to decrease as much as we can the number of symptomatic patients by treating these patients as soon as possible just after birth.
Great, thank you. So we have one more question, which is about the neutralizing antibodies. So the question is, the patients with neutralizing antibodies are not eligible for gene therapy. I think that's an important point.
Well, they're not for AAV gene therapy, and in general, there is a lot of cross immunity against the different strain of AAV. So the reality is that when it comes to newborn, we don't face so much the issue, except of course with the antibodies from the mother who are decreasing with time. But with age, we are more and more immunized against AAV, and in teenagers, it goes up to 40–50%. So the reality is that today it rules out quite a significant, except newborns, but outside newborns, it rules out quite a significant number of patients.
And is there, so that's my question, is there a cutoff that you consider, I mean, a certain titer of antibodies that would be acceptable to proceed with the gene therapy? How do you manage that?
One out of 50 in general.
Okay. Because I know some of the studies, they use as a cut of 1 in 400.
Oh, no, we are much more liberal.
Okay. I don't see any more questions. There is a new one here. What do you think about the possibility of plasmapheresis procedure before gene therapy for patients with neutralizing antibodies?
Francina, you want to take it?
I think this was one of the possibilities. No, it has been tested, but I don't know if I don't know the results of this study. But there are other studies also. One is with imlifidase, trying to reduce transiently the titers. Also, it has been tested in mice, and others is used some specific coating of the virus to try to not to be detected by the virus. But by now, we don't have um, results that show that this is working. As far as I know, I don't know, Laurent, if you.
So today there are preclinical model that shows that we could temper the immune reaction by, for instance, co-administration of nanoparticles of rapamycin and things like that. So my personal vision is that in the future, we could potentially work on getting a lower immune reaction, but we need to work on it when we give the initial dose. I'm afraid that when the initial dose has been given, it's a little bit late. But it brings back also to the question of the cost of all of this, because again, the problem is that I mean, it's today. I mean, we have to look to face the reality that the gene therapy cost today between 2, 3, or 4 millions of euros, which is a lot of for one dose. But when it comes to several doses, if we have to give it several times, it becomes a unbearable issue. So there is also this crude reality of the cost of a potential readministration.
And we need new viral vectors and probably non-viral vectors. That probably will be the solution for this problem.
Yeah, and I think one of the previous questions was about whether all patients will develop, I mean, 100% develop neutralizing antibodies after treatment, and the answer is yes, of course. So you cannot redose with the same or with a cross-reacting capsid. But what about if you do combination immunotherapy around the gene therapy, like steroids or beta depleting therapies, or what is your view on this?
So as I said, we have several, very encouraging preclinical model. To my best knowledge, there is not yet a very clear, human studies that shows that you could safely re-administer a drug. For steroids, we do it already for other reasons. Instead, our patients develop TMA and hepatic toxicity. So we anyway give steroids between. So I'm not aware, to be honest, of maybe it exists, but I'm not aware of a study that has shown that it's today feasible in humans. It's still preclinical.
Great. Okay. I don't see any more questions. Huda, I hope I didn't miss any. I don't see any more in the chat.
I believe we have answered all questions because I see a hand raised by Eugenio Zapata, but I believe we already answered his question.
Great.
And yeah, I think that's all.
Okay. So we are on time. We reached the end of the hour. Thank you all for joining, and thank you especially Laurent and Francina for these very nice presentations and for answering the questions. That was really excellent. Thank you all, and looking forward to have you join the next webinars. You can find all the schedule in the ERN website, so please join the following webinars. And thank you again for joining.
Yes, thank you everyone.
Bye-bye.
And see you again
Thank
soon.
you. Bye.
Thank you. Bye.
Bye-bye.